Question number 3. As factors affecting the rate of diffusion can be investigated in agar cubes. So straight away, we know that this question is about diffusion. The agar cubes contain an indicator called phenolphthalein. Phenolphthalein is colourless, but it turns pink in the presence of an alkali, such as sodium hydroxide solution. Cubes of agar with sides 3, 2 and 1 centimetres are placed into sodium hydroxide solution for 10 minutes. The cubes are then cut in half to see how far the sodium hydroxide solution is diffused into them. Figure 5 shows the results. And again, we've got a picture and a table to help us here. So we can see that the 1 centimetre cube size has a surface area of 6, a volume of 1, therefore a surface area to volume ratio of 6 to 1. And it's moved into the cube by 0.5 centimetres. The 2 centimetre cube size one has a surface area of 24 centimetres squared, a volume of 8 centimetres cubed, therefore a surface area to volume ratio of 3 to 1. And the distance moved into the cube is 0.5 centimetres. The 3 centimetres cube side has a surface area of 54 centimetres squared, a volume of 27 centimetres cubed. The surface area to volume ratio is left blank. And again, the diffusion distance of this one is 0.5. You could probably see this question coming. Calculate the surface area to volume ratio for the cube with a side 3 centimetres. So the surface area is 54. The volume is 27. So we have a 54 to 27 ratio. Now that's not a very easy ratio to work with. So if you want to simplify ratios, you may be able to simplify that in your head. However, if you're unsure, to simplify a ratio, what you need to do is divide each number by the smallest number. In this case, 27 is the smaller number. If we do 54 divided by 27, that gives you 2. 27 divided by 27 gives you 1. It's 2 to 1 ratio. Next question, calculate the rate of diffusion for the 10 minutes of the investigation. So we want the rate, for 10 minutes. The rate will be the what you were measuring. So in this case, it was the distance into the cube of diffusion divided by the time. So the distance was 0.5. The time was 10. 0.5 divided by 10 gives you 0.05 centimetres per minute. If you were unsure about the how to do that sum, what you needed to do, the clue is that if the unit is centimetres per minute, you need to be doing the distance divided by the time, centimetres per minute. Next question, this threw quite a few people. The question says, describe the effect of the size of the cube on the rate of diffusion. A lot of people said that as the cube got bigger, the rate of diffusion increased or decreased. If we actually look at the results, you have 0.5, 0.5, 0.5. The size of the cube had no effect. All right, so there isn't always going to be an effect on the results. So in this case, you need to read the question and the results carefully. So the size has no effect on the rate of diffusion. Okay. Next question, explain why sodium hydroxide diffuses into the cube. Again, it's worth 2 marks. A lot of people picked up 1 mark. Very few people scored 2 marks on this question. So the first mark was explaining why the sodium hydroxide diffuses into the cube. And remember that diffusion is the movement of particles from a high to low concentration. So the sodium hydroxide diffuses from an area of high concentration to an area of low concentration which is inside the cube. Now, that would get you 1 mark. The second mark is for saying how it is able to do that. Now, if it's getting through the cube, then that must mean the cube is made of a permeable substance. So, sodium hydroxide diffuses from an area of high concentration. Just check your answers. High concentrations to an area of low concentration in the cube through a permeable membrane. So the next question is a question about osmosis. So which of the following describes osmosis? A few people got this one wrong. So it does not use energy to move water, does not use energy to move glucose, uses energy to move water, uses energy to move glucose. So I think most people knew that osmosis is about water. So straight away, if you know that, then we can disregard D and B because these are about glucose. Now, the next thing is whether it requires energy or not. Osmosis is a passive process. It does not require energy. So it is A, it does not use energy to move water into cells. Next question. Animal cells will swell and burst when placed in a hypotonic solution. Explain what happens to plant cells when they are placed in a hypotonic solution. So I think this, a lot of people found this one difficult if they weren't sure what hypotonic meant. Even if you're not sure what hypotonic meant, you possibly would be able to infer what that means by this sentence here. Animal cells will swell and burst. A hypotonic solution is one that has high water concentration. If you put an animal cell in a hypotonic solution, water will move into the cell via osmosis, and the cell will burst. So for plant cells, a similar thing will happen. Water will move into the cell by osmosis. So water will osmose into an area of low concentration in the plant cell. However, our plant cell will not burst, and a few people managed to get 1 mark for this. So the plant cell will not burst because plant cells have cell walls. The plant cell will expand, become turgid. So become turgid is the same, or means to expand, but will not burst due to the cell wall, or due to the presence of the cell wall.